Hi, thank you for inviting me to speak today about approaches to enhance and accelerate the study of new HIV-related drugs in pregnancy. It is truly an honor and a pleasure to be here. Pregnant women are central to our global approach to HIV treatment. Half of persons living with HIV are women. 1.3 million women with HIV become pregnant each year, and most women will be pregnant at least once following their HIV diagnosis. Pregnancy findings can affect HIV treatment for millions of non-pregnant individuals as well, as we learned through the neural tube defect story with dolutegravir. We need to identify and provide the safest and most effective HIV treatment regimens for women and their children throughout their life course, and prevention as well. So what is the story around data for drugs in pregnancy more generally currently, not restricted to HIV? More than 90% of FDA-approved drugs have no pregnancy data. When there are data, they're delayed. Most women take at least one drug in pregnancy for which there's minimal pregnancy safety and efficacy data. The focus is understandably often on potential harm to the fetus of medications taken in pregnancy, but not taking a treatment during pregnancy can also harm mother and fetus. Pregnant women are generally excluded from research, but exclusion doesn't remove risk, rather simply shifts risk from a setting with informed consent and intensive monitoring to routine clinical settings. We've actually done a better job gathering pregnancy PK data for antiretrovirals than is the case for most other classes of drugs. However, a mean of 6 years passes between drug approval and publication of the first pregnancy PK data for drugs for which those data actually do become available. The NIH-funded PHASES group has really wonderfully articulated some key conceptual shifts that will facilitate inclusion of pregnant women in research, namely that pregnant women are a complex population rather than a vulnerable population, should be protected through research rather than from research, and should have fair inclusion in research rather than presumptive exclusion from research. We have seen an increasing call from all quarters to include pregnant women in research, and this call has accelerated over the last years with the Ebola and COVID epidemics and pandemics. I have listed here just a few of the many major initiatives that are focused on inclusion of pregnant women in research, including ongoing meetings organized by the World Health Organization and the Impact Network, which I want to highlight a little more in this talk. These WHO Impact meetings have convened a very broad group of key stakeholders, including community members and community advocates, researchers, regulators, ethicists, non-governmental organizations, industry colleagues, and funders. The Impact Network and WHO work actually started in 2019 with a workshop focusing on pregnancy pharmacology studies that brought together this broad spectrum of stakeholders. The WHO and Impact Network have since convened these stakeholders in a series of ongoing meetings with the goal of developing a framework and strategic action plan for prioritization, acceleration, and optimization of the type and timing of studies in pregnant women. 5 working groups have focused on specific challenges and potential solutions in the areas noted here, namely non-clinical or preclinical work, conducting trials in pregnant women, study design considerations, surveillance of outcomes with drugs used in pregnancy, and advocacy. And I'll talk about these in more detail. Several key messages shared by community members at the first workshop were that women, including pregnant women, are not a niche or special population. We are the population. Without inclusion in clinical trials, pregnant women will receive new drugs anyway, but in a way that is uncontrolled and unconsented. And with appropriate information and support, women should have autonomy to make the decision to participate in research themselves. I'll now talk about some of the challenges and potential solutions that have been identified through these ongoing meetings, starting with the non-clinical group. These recommendations are really still in development, and I have framed the potential solutions in a way that makes sense to me, but should not be interpreted to represent the final recommendations of the larger group. In terms of non-clinical and reproductive toxicity, or DART data, these are often not available until very late in drug development, but are generally required prior to studying new drugs in pregnancy, including before really allowing a woman to consent to stay on a study drug if she becomes pregnant in a trial. These preclinical studies are costly and challenging data to gather, particularly the animal studies. And DART study results are often difficult for clinicians to use and interpret. Some potential solutions under discussion are trying to conduct clinical studies earlier in drug development, for example, during phase 2B, selecting high priority ARVs for earlier non-clinical studies, drugs that may be game-changing for women living with HIV or for preventing HIV, and expanding new approaches and alternatives to animal models, such as ex vivo placental perfusion models, PBPK modeling, and novel in vitro experiments to screen for reprotox. There are a constellation of challenges around conducting trials in pregnant women. Currently, clinical pregnancy studies are almost always small, post-marketing, PK and limited safety studies in women who become pregnant on antiretrovirals. For example, Impact 1026 and 2026, and from the PANA network. These results have been critical and very important for women, but are delayed in relation to registration and are not always done. Some potential solutions are to move to a position where the default is for women who become pregnant in a trial, often in a phase 3 trial, are permitted to consent to stay on study drug and contribute pregnancy PK and limited safety data, unless the preclinical data raise concerns that would preclude that. To conduct small PK or limited safety studies during phase 3 in non-pregnant women, again, either in women who become pregnant on study or in a separate study that's conducted at that time. And this ideally would be done for all antiretrovirals and with available data prior to drug registration. And for some priority antiretrovirals, once the first pregnancy PK and safety data are available, to enroll pregnant women into a trial. This would probably often be in the post-registration period or potentially during phase 3 of a new priority drug. And safety endpoints would be the main of greatest interest, including birth outcomes and neonatal mortality, as well as targeted maternal and child toxicities or child growth, depending on the known toxicity profile of the drug in non-pregnant individuals. There's general agreement that there is now no need to power such studies for efficacy in terms of virologic suppression or prevention of vertical transmission, unless the pregnancy PK data raise concerns around inadequate drug exposure. The study design working group is trying to address some of the challenges related to the long delays until data are available and the somewhat costly and inefficient way in which we do studies, some of which is unavoidable, but some of which could be addressed. These studies currently are generally, again, post-registration and are often performed as a series of studies. So, for example, starting with an opportunistic pregnancy PK study, followed years later by a separate pregnancy safety study when those studies are even done in just a few instances. So some solutions that have been identified potentially are to start to plan pregnancy studies and to write those protocols earlier in the drug development phase, for example, even during phase 2, to start the planning, even if the study's not going to start till later. And as part of that, to establish recommended minimum data and sample collection guidance and template protocols, and to simplify and streamline follow-up and case report forms. Another attractive approach is stepwise enrollment, starting with enrolling women who are in the late 3rd trimester, followed by enrolling women who are earlier in the 3rd trimester, and then women in the 2nd trimester, with external safety monitoring prior to moving from one step to another. And considering alternative designs, such as seamless designs, for example, integrating pregnancy PK, followed by safety protocol endpoints into one protocol rather than separate protocols, considering pregnancy platform trials, and emulated target trials using observational data when those data are available. The surveillance work group has been discussing the known challenges of surveillance, including unknown denominators, reporting bias, and underreporting that can plague voluntary registries. The existence of only a few data sources of pregnancy surveillance from a limited number of settings, most of them in North America and Europe, and incomplete or inaccurate data that can be found in routine medical records when those are used for surveillance. Some possible solutions include establishing a few surveillance centers of excellence in areas with high HIV prevalence and advocating for funding thereof, potentially even requiring post-marketing surveillance for new drugs that are used widely, and harmonizing data collection procedures. Also, leveraging and improving the quality of electronic health records and finding ways to link maternal and health data that are maternal and infant routine health data. The advocacy group has been focused on some of the norms around research and pregnancy in which pregnant women are by default excluded from research because they were previously listed as a vulnerable population. And researchers generally need to justify why pregnant women should be included to one another and to regulators and IRBs. Another major challenge is insufficient communication with and involvement of affected communities in research from the outset, and disincentives for industry to do research in pregnancy, including liability, potential delays, and costs thereof. A key solution is engaging community throughout this process in a meaningful way and identifying the main messages and advocacy opportunities across a range of stakeholders. Advocating to stakeholders to adopt a position of requiring justification of exclusion of pregnant women from research, developing a multi-stakeholder call to action, and hopefully creating an accountability platform. This slide summarizes again some of the key potential approaches under consideration for accelerating research in pregnancy, including obtaining non-clinical data earlier in drug development, permitting women who become pregnant in trials to consent to stay on study and on drug and to contribute PK and safety data unless there is a reason not to from the preclinical data, for example. And for priority drugs, planning dedicated pregnancy PK or larger safety studies to start post-registration or during phase 3. Also expanding and improving post-marketing surveillance for rare outcomes and engaging community throughout. Collection of pregnancy data is currently planned for a subset of these newer drugs that are in various phases of development. The ones with some pregnancy study planned are indicated in dark blue, and these studies are in relatively small opportunistic studies, many of them post-registration, with the exception of the dapivirine ring, which is being intentionally studied in pregnancy and lactation trials. One encouraging development is that women who become pregnant in trials of long-acting drugs, such as cabotegravir, rilpivirine, islatravir, lenacapavir, will be able to consent to stay on study drug and contribute pregnancy PK and safety data. I think we'd all agree that long-acting agents are very important new drugs for both the prevention and treatment of HIV. And it's important to remember that even if a woman who becomes pregnant stops a long-acting agent in the first trimester, the drug will likely be present through delivery. We have almost no human pregnancy or lactation data for long-acting agents yet. In conclusion, women deserve high-quality evidence for medications that they will use throughout their life course to help inform their care and decision-making, including during pregnancy and lactation. Thank you for listening, and thanks to the many women who take part in pregnancy research, as well as to Drs. Penizato and Abrams and the many other individuals listed here for their leadership in this current effort.